Translation has numerous steps, and the activity would require each team to have a DNA kit, a protein kit, and a tRNA kit. Therefore, several teams trying to do this simultaneously is not practical and may result in reinforced misconceptions. We recommend a teacher-led participatory demonstration of translation. Before leading this demonstration, prepare a mutated beta tRNA kit and also a mutated beta mRNA strand. See the tRNA booklet number 1 for these instructions. You will need to have 6 amino acids for the mutated beta protein and the ribosome mat as well. During this activity, make sure that the students participate in the translation process instead of just watching. Common student mistakes can be useful for correcting misconceptions. Read the tRNA teacher guide for more information on preparation, classroom tips, and key concepts. There's room back here. This is the grand finale. We actually are going to be able to make a protein out of all these steps that we've done. So, do you recognize this little object? It's the ribosome, right? And what belongs here?
The mRNA.
Okay, the mRNA. And so, does somebody want to position the mRNA? Let's just try it out. Don't worry, we'll guide you if you don't know where exactly it goes. This, you can see which end is the AUG, which has got to be the beginning, right? So can you find the beginning?
Here.
Okay, so you want to just move it over? Thanks.
Like,
Yep. Great, okay. Nice. We want to flip these up. What happens is, this section that's here is going to be able to hold tRNA when it comes in. tRNA doesn't grab on out here. It's really lucky it doesn't happen that way. So this is really the first site. And I don't know if you learned the letters. This is E, P, A. And if you've, E is for, I know, it's leaving.
exit.
Exit, okay. So, and P and A have to do with the bonds that are formed. So I just remember EPA because that's the name of an organization. But anyway, here we are. We're clamped in here. We need to find a MET, and you can see that I put on the tRNA the name of the amino acid and also the RNA codon that you would find if you looked it up in a chart. Okay, so I'd like to hand these out to a couple people and have you pick your amino acid and put it on. I'd like to try that. Got one? I only have 6 right now, so try it out. You'll get a chance, I'm sure, later on to try it again. So here are some amino acids. And when I was designing this, it was really fun for me to discover that the amino acid is always attached to the tRNA by the acid group. So in other words, it was really easy to make this design, right, that it would fit on there. But we have to make sure that the right ones go together. And there's a protein that does this, in case you're wondering, a protein that knows to put that tRNA always with a particular amino acid. So would you find your amino acid and pretend you're the synthase that's going to add it? That'd be great. A good protein adds it. There's somebody that should not be adding a amino acid. Yes, okay. Didn't realize who I handed it to. That's good. All right. So just randomly, I'm going to ask for you to come in here and put this, try to add it here to the ribosome that's holding the mRNA. It would probably, well, somewhere in these positions. Let's see if it fits. Just see if it fits. And it's got to be either in this position or this position, okay? So I asked you to do the impossible thing. It doesn't fit, right? It's not going to bind here. It's not going to bind here. So I know that in many animations, they just show the right amino acid on the right tRNA coming right in, and it just binds. But there is this sorting mechanism that happens on the ribosome. So it can't, the tRNA just doesn't read it. It ends up having to bind by itself. So any of us that can read, are you the MET?
Yeah.
Okay, so you want to come in and bind here? That'd be great. Yeah. It's good, okay. All right, so does anybody know that they belong next? All right, so we'll let you come in here. I just wanted to make the point that they do try it by trial and error. Okay, so now what we've got is we've got this grand moment where there's a possibility of making the protein chain. But I'm looking at this, and will these two bind?
No.
No way, okay. This is amino end and amino end. So is life hopeless? No, it turns out that the ribosome is really very special, and it does cause a reaction to happen. It takes off this and puts it on here. It takes energy, so that you have to give up energy in order to make this first bond. And then what happens after that, I'll do this next step. And after this, we'll have the next person come in and they follow along and do all the steps. So remember that this one that was in this position here goes off and comes on the end. And look, which amino acid is it? It's a MET, right? So the MET is going to stay on the end there, and we're going to move over 3, like that. And then what happens here is this gets exited, like that. And we're ready for the next one. So if you have the next tRNA, would you bring it in and then do all the steps? We'll coach you if you need help, okay? Great. All right, so now we're established, and now there has to be a reaction.
Does it lose both or does it lose?
Yes, good question. And look at this, the MET's still staying on the end. That's how it works. Okay, and then what happens next? Shift over. Everything shifts over. It will not exit until it hits the exit spot. We can make sound effects now if we wanted to, right? And then,
Break off these.
no, that stays on. This is our messenger RNA. It's going to stay intact. And then we end up breaking the hydrogen bonds, which are really easy, right? Okay. All right. And these, anybody have an idea what happens to these? These tRNA? Guesses? Are they discarded?
No, they're saved.
Yeah. These are like taxis. I think of tRNA as being taxis, and they can be reused and come back again. All right, so we've got our next actor. It's a tRNA bringing in a glue. All right. Takes energy. Pop. All right. Moves along. And then exits. Stage left. All right. Nice. We're getting a really cool protein here. Good. Yep. Got along 3. Yep. Gets. All right. Can you see? Okay. Good. All right. Nice. We're getting really. Do you know how fast this is? Normally, we are like miles and miles slow. This really takes only a few seconds, less than, to make this protein. The cell is much better than us, for sure. All right, now you come to a great point. So when this gets added, actually, when I did the design, I had to make it look like this because there wasn't any other way to make just one single mold for that. So it actually is a protein. It's really interesting. And it does come in here, and it's not exactly like the rest of the tRNAs. But what it does is terminate the entire thing. So it makes everything, it adds a hydrogen here. So this will come off. And then what we'll do is you can make it fall off. So that's what happens. This falls off, and everything else gets released. So it releases from the ribosome right there, and we've got our protein that's separated.
The MIT Edgerton Center presents Translation, to be used with the MIT Edgerton Center DNA, RNA, protein, and tRNA sets. The ribosome uses transfer RNA to read the messenger RNA and make a protein. This process is called translation. Transfer RNA, or tRNA molecules, carry amino acids to the ribosome. Each tRNA carries one specific amino acid. When a tRNA has an amino acid attached, it is called a loaded tRNA. Load each of your tRNA with the amino acid on the sticker. After leaving the nucleus, the mRNA travels to a ribosome. The mRNA always starts with A, U, G. The A should start with a phosphate group. The AUG of the mRNA strand is placed on the start section of the ribosome. Place the loaded MET AUG tRNA in the P site of the ribosome. MET is always the first amino acid in every protein chain. The tRNA should bind to the mRNA with all nucleotides pairing correctly. Place the loaded pro-CCC tRNA in the A site of the ribosome. It should bind to the mRNA with all nucleotides pairing correctly. Remove the MET amino acid from its tRNA and attach it to the pro amino acid to make a peptide bond, as shown with the arrow. Slide the mRNA over by 3 nucleotides. Detach the empty tRNA from the mRNA by pinching and move it into the E site. E is for exit. The tRNA is then released to be reloaded and used again. Place the loaded SER UCU tRNA in the A site of the ribosome. It should bind to the mRNA with all nucleotides pairing correctly. Remove the MET pro chain from its tRNA and attach it to the SER amino acid as shown with the arrow. Slide the mRNA over by 3 nucleotides. Detach the empty tRNA from the mRNA by pinching and move it into the E site. The tRNA is released to be reloaded. Continue adding the correct tRNA one by one to the A site, moving the protein chain to the right to add the next amino acid. Remember to slide the mRNA over and detach the empty tRNA. The order of the amino acids is determined by the order of the mRNA nucleotides. Every group of 3 nucleotides is a codon and codes for a specific amino acid. The mRNA codon must pair exactly with the tRNA anticodon for the amino acid to be added to the protein. The stop tRNA is actually not a tRNA molecule. It is a protein molecule called a release factor. Place the release factor in the A site of the ribosome. It should bind to the mRNA with all nucleotides pairing correctly. When the release factor binds to the stop codon, all the molecules are released from the ribosome. Remove the protein chain from the tRNA. Remove the empty tRNA, the release factor, and the mRNA from the ribosome. The protein created here is a beta-mutated channel protein chain. Translation completes the central dogma of molecular biology.
So does anybody have any questions so far about how or what if scenarios maybe? Thinking about it?
Where does the mRNA go afterwards?
I was wondering if somebody was going to ask that question. It happens that if you're looking at as a microscopist and you're looking in the cell, many times you'll see really closely, you can actually see lots of ribosomes on the same long messenger RNA. So they seem to like jump on in different places, and so many times you'll have, it looks like many ribosomes are attached at the same time. And the story, it would make great research projects, is figuring out how long these will last and what happens to them and things like that. So Those are still a lot of research questions going on. So we have our messenger RNA, and we have our protein here. Let's do the last bit, and this is that this would fold up. And we have here, do we have a mutation or not? Do you recognize this? Yes, we do, right? We have a 2, and then 3, and then 2. So when this wraps up, there's a possibility we'll be in trouble when we try to get it into the membrane. There we go. We have this threonine here where it should be a hydrophobic one. And actually, I chose this example. I was going to go over here and get this little bit of a membrane here. Because this is an example of a genetic disease, and maybe you studied it in biology. Do you remember a genetic disease that has problems with their cell membranes?
Sickle.
I'm hearing a lot of C's. Not sickle cell, because that's a good thinking about. That is a genetic disease, but it's not the one that has to do with cell membranes.
Is it cystofibrosis?
Cystic fibrosis.
Fibrosis.
Yeah. So I was hearing the sisses. That's great. You pulled that out. So basically, what's happening is it is handed down genetically, and it's the most common disease, genetic disease for Caucasians. So I decided to include a teacher note right now while we're talking about channel proteins because I found it's really useful to be able to explain some terms such as gene, trait, and protein at this moment. Genes and traits are usually referred to when we're talking about heredity, but now we're adding proteins to the mix, and our protein is a channel protein, and it's a good example. So we understand a lot more about cystic fibrosis through a lot of research. And that research shows us that there is a gene that produces a defective protein, and it's a channel protein, and that's what causes the disease. And so now we can use our understanding about the molecular biology to go a little bit deeper. All right, so let's imagine what's happening with genes and traits and proteins. So let's say that we know that a parent and another parent here can contribute their genes to an individual here. So we will have a pair of genes for a channel protein in an individual. And what I'm showing you is with my open palms that this is a working protein, and this gene can make a protein which will be perfectly normal. But in the case when I show my hands like this, we're indicating that this is a gene that will make a defective protein, as in cystic fibrosis. So let's think again. So this time an individual inherits a working gene that makes a working protein, and this gene over here works, but it makes a defective protein. Well, what happens is a person may never know that they're a carrier in this case because the cell can use this gene to make a working protein. And this one may still make proteins, but they may not be functional. So the real problem always happens in cystic fibrosis when an individual inherits, of course, 2 defective genes, and there is no other go-to way of making working proteins. So it helps to understand that traits are not just things that people talk about that have to do with eye color or hair color. They involve proteins, and proteins are the ones that make for these traits. And in this case, this channel protein is a good example of how you can teach about this.